Hello everyone, we are Group 2. In this video, we will talk about minor histocompatibility antigens, in short, MIHA. So, what is MIHA? In the human body, all cells need to identify that they belong to the same body. They do this by presenting two molecules specific to a person on the cell membrane. The two molecules are MHC, major histocompatibility complex, and MIHA, minor histocompatibility antigen. MIHA is a small peptides which are made inside a cell. As you know, proteins are made through the process of central dogma, and then some of proteins are broken down and become MIHA. Again, by presenting outside world what are being made in the cell defends itself from the immune system. Cells from different people may have different genes or DNA, so this results in different MIHAs.
Different MIHAs could cause graft rejection. In the following studies, you will see how skin grafts affect recipient mice without immunosuppressive treatment. When the donor and recipient are syngenetic, that is, they are identical at MHC and MIHA, there is no problem. Syngenetic donor and recipient include identical twins. When skin graft is transplanted between mice whose MHC and MIHA are different, rapid graft rejection occurs, and the skin graft dies within 10 days. When the donor and recipient are identical at MHC, but different at MIHA, the graft from the donor is rejected slowly, and the skin graft dies within 60 days. The results show that different MIHA could cause graft rejection. Stop the video and look at the figures carefully if you need to.
As the name suggests, when transplanting organs or tissues, major histocompatibility complex is a big deal. However, minor histocompatibility antigen does play a role in graft rejection, but its significance is not much as it is for MHC.
The cancer of white blood cell. White blood cells are made from the hematopoietic stem cell, which resides in the bone marrow. During the differentiation process, hematopoietic stem cells go through various stages of progenitor cells to eventually become blood cells, including white blood cells. However, it is believed that some genetic abnormality occurs in the progenitor cell before they mature into white blood cells, which will cause leukemia. To cure leukemia, hematopoietic stem cell transplantation, or HSCT, may be considered as a treatment plan. HSCT is the transplantation of the hematopoietic stem cell, and it has two benefits. First, healthy hematopoietic stem cells are the source of healthy blood cells. Additionally, T cells from the donor hematopoietic stem cells may target and destroy cancerous white blood cells in the recipient blood. This is called graft versus leukemia effect. The donation must come from a person whose human leukocyte type antigen, or HLA, human version of MHC, is matched with that of recipients. This is to avoid graft rejection, as we discussed in the previous slide. Often donation comes from the sibling because there is a higher chance of a match between the two HLA types. Even if we have matched HLA types minor histocompatibility antigen, MIHA may differ between donor and patient due to their genetic polymorphisms. And this will cause graft versus host disease because T cells from the donor recognize patient cells as non-self and attack them. Stem cell transplantation expects graft versus leukemia effect. However, because MIHA in each person is almost inevitably different, there is always a certain level of graft versus host disease. Medical practitioners need to balance these two important factors and do their best to help the patients.